Look at the individuals in this photograph performing manual harvesting. For most of history, farming looked exactly like this. Third, they cross-hybridize those selected parents. Fourth, they select and test the superior recombinants. And finally, they test, release, and commercialize the new cultivars. These 5 steps form the basic timeline of crop development. But to secure top marks on your exam, we have to look closely at what happens during Step 3 and Step 4. This is where agricultural scientists use specific genetic tools to physically hack plant biology. Let's examine Step 3, cross-hybridization. To guarantee we combine traits from 2 specific parents without accidental interference, we must prevent self-pollination. We do this through emasculation, physically removing the anthers from a bisexual flower before they mature. Immediately after, we cover the stigma with a butter paper bag, a necessary precaution known as bagging. Depending on the genetic distance between the chosen parent plants, breeders operate across 4 different levels. Intravarietal crosses mix plants of the exact same variety. Intervarietal crosses mix 2 different varieties of the same species. Interspecific crosses blend distinct species within a genus, and intergeneric crosses combine entirely different plant genera. The ultimate goal of crossing these distinct plants is to achieve heterosis, commonly called hybrid vigor. This is the biological phenomenon where the F1 hybrid offspring physically outperforms both of its homozygous parents in yield, growth rate, and disease resistance. The dominance hypothesis explains exactly why this superior performance occurs. If we cross a weak parent carrying homozygous recessive alleles with another weak parent carrying different recessive alleles, the resulting F1 hybrid inherits dominant favorable alleles from both sides. In the offspring, these dominant alleles successfully mask the harmful recessive ones. By intentionally hiding those deleterious traits, breeders use hybridization as a deliberate genetic strategy to guarantee a physically robust, hyper-vigorous crop. Sometimes breeders bypass normal genetic inheritance entirely, using a technique called polyploidy. This method forces a plant's somatic cells to retain more than the standard 2 sets of homologous chromosomes, intentionally breaking biological rules to produce massive physical changes. The first category is autoploidy, where chromosome sets multiply within a single species. This creates what botanists call the gigas effect, yielding larger cells, broader leaves, and often sterile, seedless commercial fruits, like giant watermelons. The second category is alloploidy. Here, sets of chromosomes from 2 entirely different species merge. A required board exam example is triticale. Scientists hybridized wheat and rye, then doubled the resulting chromosomes to restore the fertility of the offspring, successfully creating a completely man-made cereal grain. While hybridization allows breeders to mix existing traits, polyploidy gives scientists the power to restore fertility to sterile hybrids and invent entirely new plant architectures from scratch. Even when high-yielding crops produce enough sheer caloric volume to feed a population, they often lack essential vitamins and minerals. This dynamic creates a severe public health crisis known as hidden hunger. To combat this, agricultural scientists rely on biofortification. They breed crops to contain much higher levels of vitamins, minerals, healthy fats, and proteins. A prominent example is golden rice. It is genetically enriched with beta-carotene, or provitamin A, specifically to prevent deficiency-induced blindness in developing nations. Here is an excerpt from your textbook detailing another critical example sample. Atlas 66 is a specific variety of wheat containing extremely high protein content. Scientists use it directly as a donor parent to improve the nutritional profile of standard cultivated wheat. Let's lock these concepts in. Pause the video right now and test your active recall by matching these three plants, golden rice, Atlas 66, and triticale, to their specific agricultural traits. Let's verify. Golden rice is enriched with vitamin A. Atlas 66 is your high-protein wheat, and triticale is the man-made cereal cross of wheat and rye. Memorizing these specific pairings guarantees you will be able to answer the direct application questions on your test. Let's apply everything we've covered to a classic 5-mark WBCHSE board question. Define plant breeding. Name one biofortified crop and explain its significance. We start our answer with the precise definition: the purposeful manipulation of plant species to create desired types. Next, we name golden rice, and we specifically explain that it is biofortified with beta-carotene to fight vitamin A deficiency blindness. To pull this entire chapter together into a single review frame, here is your exam cheat sheet. Note the physical mechanism of emasculation to prevent self-pollination, alongside the genetic goal of heterosis, which is driven by the dominance hypothesis. Complete your study list with the alloploidy example of triticale and the specific biofortification examples of Atlas 66 and golden rice. Take a screenshot of this frame. Mastering this exact vocabulary and these specific crop pairings ensures you have the exact toolkit needed to secure top marks in your Enhancement of Food Production unit.